The title of our presentation is The Contribution of Neuroendocrine Factors on Response Inhibition in Female Rats. It's going to be presented by myself, Samson Reed, Caitlin Davis, and Bree Rasmussen. So we're just going to walk you through background and purpose, and that'll be done by myself. And then the methods and procedures used in the experiment will be done by Caitlin, and then our results and findings will be presented by Bree. So our first point is that addiction negatively impacts a person's behavior and brain function. And a critical component to the stopping of addiction is the ability to inhibit a previously learned behavior. So previously in our lab, we've showed that acute stress will significantly decrease a response inhibition. But we did this with male and female rats, and the female rats showed a high variability in their response inhibition. And we thought this might be due to their menstruation cycle and the way that works with estrogen. So their estrogen is going to fluctuate throughout their menstruation cycle, and because they have a shorter lifespan, their menstruation cycle is also shorter. It's about 5 days, so it's harder to control when you're testing it against males and not controlling specifically for it. So the purpose of our present study was to investigate the relationship between estrogen and stress and how it impacts response inhibition. One method that we used to stop the behavioral response is through extinction training. And that is meant to weaken the association between the behavior and reward. Basically, as humans, or like any species really, we're constantly either activating or inhibiting responses. And so the extinction training is just a way to help the subject inhibit that response by weakening the association between that behavior and the reward that they're getting from that behavior. And spontaneous recovery is when the behavior that was extinguished returns after some amount of time, and that is supposed to measure behavioral inhibition later. So a good example of spontaneous recovery would be if you were going to go on a diet. So you kind of teach yourself that you need to be eating salads over this period of time, and you're giving up, say, pizza. And so after a couple weeks of eating just salad and just healthy foods, you might get like a craving for pizza, and that would be the spontaneous recovery of, like craving foods that you're not supposed to have.
Okay, so our subjects were 16 ovary-activated female rats. And ovary-activated just means that their ovaries have been removed, and so there's no naturally fluctuating levels of estrogen in their body. So it just creates baseline level for all the rats. And so in order to manipulate the estrogen, we administered subcutaneous implants filled with either 10% estradiol and cholesterol, or 10% cholesterol as a control group. And so after these implants were put in, over time they slowly release. So that's how we just manipulated the estrogen levels. And then to test stress, immediately prior to the spontaneous recovery test, half of each treatment group received 30-minute acute restraint stress. And so we use these clear restraining tubes, and we place the rat inside, and this doesn't cause any harm to the rat, but it just causes stress, and it causes the physiological stress response, which includes an increase in corticosterone levels. And so this is the touchscreen chamber that we use during training. And so we have the chamber where we place the rat, and then here is the touch screen, and the stimulus is the little white square. And so every time the rat touched the stimulus, it received a food reward pellet from over here. And so for acquisition, there are 100 presentations of the conditioned stimulus for each session. And so if the rat touched the stimulus, it received the reward pellet. And the acquisition criterion was 85% conditioned responses within a single session. And then for extinction, 60 trials were used per session. And this was where no reward was given for touching the conditioned response. And so the extinction criterion was 24% conditioned response or lower in a single session. And then spontaneous recovery was 2 weeks after the rats reached extinction criterion. And so after they reached extinction, there was a 2-week retention period where no training was done. And then after the 2 weeks, there was 1 day of testing to test the spontaneous recovery.
This first figure, figure 1, shows the number of sessions to meet criteria for both estrogen-treated rats and then cholesterol-treated rats. There appeared to be no difference between the number of sessions it took for the estrogen-treated rats and cholesterol-treated rats to reach the criteria, and it took like about 2 to 3 days for each group. For figure 2 right here, you can see an acquisition curve, which is basically the number or percentage of conditioned responses for the 10 trial blocks. The percentage of conditioned responses increases over time, and as you can see with the trends, there was not really a difference in the touches between the estrogen-treated rats and cholesterol-treated rats. Figure 3 shows the extinction period over 3 days, which is separated by these black lines. This x-axis still shows the 10 trial blocks, and this y-axis still shows the percentage of conditioned responses. The extinction starts off pretty high at 100%, and then it decreases over time and ends at around like 20%. Then figure 4 shows the spontaneous recovery, this time over 6 blocks, and the same y-axis with the percentage of conditioned responses. The spontaneous recovery is the lowest in the cholesterol-treated rats that were not stressed, and the estrogen-treated rats, both stressed and no stressed, in the red are showing generally the highest spontaneous recovery. In conclusion, the cholesterol-treated and estrogen-treated rats both acquired the touchscreen conditioned response at the same rate. Both stressed and not stressed estrogen-treated rats showed a higher spontaneous recovery compared to the cholesterol-treated rats. Then the cholesterol-treated rats that were not stressed showed the lowest spontaneous recovery compared to the rest of the groups. Overall, these findings suggest that both estrogen and stress induce changes in response inhibition, and then additional subjects will be tested to support these findings.